Sure. Let's start from the heart. Heart, left systemic aorta. Left systemic aorta will get down in the form of dorsal aorta. Okay. So dorsal aorta enters into renal artery. Okay. Renal artery opens into branched in the form of renal arterioles. Renal arteriole branched into afferent arteriole. Afferent arteriole form glomerulus. Okay, afferent arteriole form glomerulus. And glomerulus will come out in the form of efferent arteriole. Okay, efferent arteriole. Then from the efferent arteriole, that above on the PCT and DCT, what is there? Peritubular net. Then above on the descending and ascending limb of Henle's loop, vasa recta. Yeah. Okay, then after that, all they get open into renal venule. The renal venule opens into renal vein, and renal vein opens into post-caval vein. Post-caval vein opens into heart. So this is the journey from heart to heart. Actually, we need to say here left ventricle of the heart to right atrium of the heart. Okay, so this is the sequence which we are completed in the earlier class. Okay, so let me show you the visual for the sequence also. Let me show you the visual. What we said, from the left ventricle, the left systemic aorta is coming down. The left systemic aorta will get down in the form of dorsal aorta, then renal artery. Renal artery enters into the kidney, and in the kidney, renal arteriole. Renal arteriole is going to form afferent arteriole, and that form the glomerulus. And glomerulus will come out in the form of efferent arteriole, and then after we made some peritubular net. Above on the PCT and DCT, we made some peritubular net, then vasa recta, and vasa recta will get open into renal venule, renal venule into the renal vein into the post-caval vein, and post-caval vein that opens into the right atrium of the heart. So this is a complete journey. This is a complete journey. You people need to understand this is left ventricle, left systemic aorta, and dorsal aorta, then renal artery, renal arteriole, and afferent arteriole, and this is glomerulus, and efferent arteriole, then peritubular net, vasa recta. Peritubular net, vasa recta opens into the renal venule, renal vein, and then that opening into inferior vena cava. Then finally we came back to the right atrium. So it is clear that how much blood is coming out from the heart? How much blood is coming out that we discussed in the form of cardiac output? You remember in the previous chapter we discussed in the form of cardiac output, isn't it? Yeah. So now you see directly answer how much blood is coming. 5040 ml cardiac output equal to stroke volume into number of heartbeats per minute. So where does this is going? It is going to all the body parts. So how much to the kidney is going, sir? That's the question here. How much to the kidney? That we can say 1/5 of the volume of the particular blood pumped out by ventricle. 1/5 volume of the blood. 1/5 volume of the blood pumped out by that ventricle is going to be enters into the kidney. Enters into the kidney. So can you say approximately that total is coming out 5040? In that we are saying 1/5 is coming to the kidney. Kidney is also a body part. Yes. How much? 1/5. 1/5. That means here out of 5 parts, 1 part. That means it is going to be nearly 1100 ml to 1200 ml of the kidney enters through in this particular way. In what way? Renal artery 1100 to 1200 ml of the kidney entering into the renal artery, renal arteriole, glomerulus, likewise, whatever it is. Just keep this in mind. We are going with another sequence here, another sequence, the sequence of Bowman's capsule. When we start the story from Bowman's capsule opens into proximal convoluted tubule, PCT, isn't it? Proximal convoluted tubule opens into descending limb of Henle's loop. Descending limb of Henle's loop. Descending limb of Henle's loop opens into ascending limb of Henle's loop. Ascending limb of Henle's loop. Okay. Ascending limb of Henle's loop opens into distal convoluted tubule. Distal convoluted tubule opens into initial collecting duct. Initial collecting duct. Okay. Then initial collecting duct opens into straight collecting duct. Actually not mentioned in the NCERT, but writing here. Ducts of straight collecting duct. Then when it comes into ducts of Bellini. Ducts of Bellini. And all the ducts of Bellini opens at renal calyces. All them opens into renal calyces. Renal calyces opens into pelvis. Pelvis into ureter. Then ureters into urinary bladder. So, okay, afterwards that is will be eliminated out by urine. If it is male, we can say urethra. If it is female, we call it as vestibule. In male, urinogenital aperture, urethra. In female, vestibule. Whatever. That means here, if you see here, this is Bowman's capsule, proximal convoluted tubule, descending limb and ascending limb, and distal convoluted tubule, and initial collecting duct, and all the initial collecting duct opens into the straight collecting duct, and they are opening into the ducts of Bellini. They are opening into the ducts of Bellini, and all the ducts of Bellini opens at renal pelvis. Renal pelvis opens into the ureter. Ureter opens into the urinary bladder. So this is the story. Okay, so let me show you. This is Bowman's capsule, and this is proximal convoluted tubule, descending limb of Henle's loop, and ascending limb of Henle's loop, distal convoluted tubule, initial collecting duct. There you can see the same here even. That means here, the same diagram I am going to draw here also. Bowman's capsule, PCT, Henle's loop, just for your vision, were shown. And ducts of Bellini, here it is, renal calyces. All these are the calyces. Calyces opens into the pelvis. Pelvis opens into the ureter. Ureter opens into the urinary bladder. Okay, so two different sequences I made it for you. One is the blood vascular system, and other is the excretory system. Okay, now the third sequence which we are going to do is making the topic. That means excretory system, urine formation, which is a major part of this particular topic, depends upon this particular area. For that, let me show you what is happening here. What is this? Bowman's capsule. And what is this? Afferent arteriole enters into the Bowman's capsule. Okay, afferent arteriole which enters into the Bowman's capsule. Okay, then this is, what is this? Glomerulus. If I ask you glomerulus, from where are you coming? Yes, from where it is coming? For that only we have to see the first sequence. Glomerulus, glomerulus. From where are you coming? I am coming from the heart. Left ventricle. I am coming from. What are you bringing? I am bringing blood. How much? 1100 ml to 1200 ml. That means I am giving priority to you. Out of the total cardiac output, how much blood brought out towards the kidney? 1100 to 1200 ml. Okay, the blood is moving. So why the blood came here? Simple, we can say why the blood came here to bring out excretory wastes here. Okay. So we call this process as ultrafiltration. We say this process as ultrafiltration. Now the question raises here, how much amount of that particular blood excretory wastes are going to be eliminated into the neck of Bowman's capsule? Then if it all continued over, this is the excretory waste. Just understand this is the excretory waste. Where it will go? Yeah, that we need to ask the second sequence. If it is there anything in Bowman's capsule, where it will go? Okay, end I am asking here. End I am asking here. Yeah, just simply we can say here, we came here and dumped the excretory wastes. That's the end of the story. That means we need to bring out the sequence like this. So from the heart, what we have to say here, left ventricle, left systemic aorta, dorsal aorta, renal artery, afferent arterial, glomerulus, excretory materials are filtered in the Bowman's capsule and they go in the till the urinary bladder, PCD descending and ascending Henley's loop, DCD collecting duct, straight collecting duct, Bellini, the calyces, pelvis, ureter, urinary bladder. This is a small story. Then if it is this much smaller, so why that heading urine formation? Then why the various kinds of techniques? So simply we can say here, the Bowman's capsule and glomerulus, the relation, that means filtration, which is happening in between at the time when they came for dumping of, for what they came here for dumping of excretory wastes only. But along with the excretory wastes, we found some issues. So actually we came here to dump the excretory wastes, isn't it? But glucose is dumped over, amino acids, vitamins, and minerals. That means came here to dump glucose, amino acid, vitamins, minerals, large amount of water is dumped over in the dustbin. If you leave, where all will go? Yeah, that's why there a particular mechanism we require over. So simply we can say here, usually I use it to say an example here. This is your house. Okay. So there nearby your house, there is a dustbin. You came here with a small box, carrying the small box. Don't look towards the painting. That carrying a small box, you came with the dust of your house, that you came toward the dustbin. Okay. So this is a municipality dustbin. And what happened here? When you are dumping the dust into the dustbin, along with that, some valuables you lost. Your ring, your chain, likewise. Some valuables you lost in the particular bin. Okay, then after GHMC people came and the municipal people came and taking this dustbin towards the dumping yard. Okay, towards the dumping yard. So now you are in the van. You are in that particular van where that carrying that particular dustbin, because you came here for to dump the dust, but also along with that, what happened? Your gold ring, your gold chain. I am not talking your books are dumped. Unfortunately, if I say, do you run behind them? No. So that's why I'm saying people feel more than valuable than a book is gold chain, gold ring, diamond chain, diamond ring. Let's make it bigger. Okay. So if you leave, what happens? They go to the main dumping yard. Remember, once they go to the main dumping yard, you don't get that valuables. That's why in the journey itself, you have to take all of them. Okay. So that's what I am saying here. That's what I am saying here. What I am saying. Okay. You dumped excretory wastes here, but along with the excretory wastes, what has also happened here? Various kinds of valuable glucose, amino acid, vitamin, water, all they get dumped. If you leave, where it will go? Yeah. So what is your task then? Yeah, you need to absorb all this particular material. You might have observed over the glomerulus came for dumping. After that, they are not going. They are coming back again. Why? Can you give the reason? Yeah, valuables are there. That's why blood vessels are not going from there. They came back. Peritubular network and vasa recta. This is called conceptual understanding. If you understand like this, let's try to make some sequences here before we enter into the topic. Let's try to make some sequences. Okay. Then Henley's loop. Along with the Henley's loop, I am going to draw an nephron to you. This is Bowman's capsule, proximal convoluted tubule, and descending limb, ascending limb of Henley's loop. And I already said over cortical fluid and medullary fluid. That issues I said already. Let me use another color. Let me use the black. Bowman's capsule, proximal convoluted tubule, descending and ascending limb of Henley's loop, distal convoluted tubule, and initial collecting duct. We know where this way goes. What is the end of this road? Urinary bladder. Who did the mistake actually? Afferent arteriole. Enter inside. What is this red one? Glomerulus. For what they came here? To dump the wastes. Okay. But along with the wastes, what is also happened? So, as I already said to you yesterday, that this is cortex and this is medulla. The positions. Here, whatever the fluid present, we can say cortical fluid. And whatever the fluid present here, we can say medullary fluid. We'll discuss later, but back again. But for this understanding, what is the arrow mark meaning? I'm writing here. Glucose is absorbed into. Yeah, you answer it. Just I said it over that whatever the fluid present here, what do you call it as? Cortical fluid. Okay. So then that particular glucose who came here, that particular glucose who came into the cortical fluid, is there any blood vessel taking them? Yes. Who is this? Peritubular net. Okay. So where it is going along with the vasa recta? Very good. Going to the renal vein. Inferior vena cava. Chalo. Let's write about the glucose journey here. Journey of glucose. That you will be able to understand. Okay. Journey of glucose. The glucose we take example as high threshold substances. That means the substances which after dumping, that means after dumping, which we should not leave towards the urinary bladder, which we require. We want this. We want this term completely. What do you say? The kind of substances, high threshold. As an high threshold substance, let's make a sequence. Okay. So glucose. Actually, where the story starts, I started in the heart, left ventricle of the heart. Chalo, do help. Left systemic aorta, then dorsal aorta, then renal artery, then renal arteriole, then afferent arteriole. Okay, what happened? Glomerulus. Then from there, where it get entered? It was filtered actually. See this, that's what I'm talking about. Glucose came like this and filtered here. Where? Into the Bowman's capsule. Okay. So digest the topic. So after that, where they came? Into the proximal convoluted tubule. Where they came? Into the proximal convoluted tubule. Okay. From the proximal convoluted tubule, where they get reached? No, they are absorbed. Cortical fluid. You see here the glucose journey. They are absorbed into the cortical fluid. Who is taken from there? Peritubular net. Okay. So from there, where you gone? Renal venule. Then renal vein. Then inferior vena cava reached to the heart. Okay, safe journey. So that means yes. పెట్టేస్తాను yesterday's and today's. Okay, so that means practice of the sequence is very important. This is the soul of the topic. As we can't say like that a regular animal kingdom structural organization like a line by line explanation is not like that. You need to understand the soul of the topic first of all. When you understand the soul, what is going to happen when it comes in mind, then only you will understand what exactly happening. Means here in the first sequence I said over, there is a blood vessel, like we get started from the heart, then glomerulus came, and after that their return journey. Okay, next, and next sequence in the nephron sequence we made it out. Nephron sequence, Bowman's capsules to urinary bladder. Then actual topic we get entered here. The glomerulus came here for filtration, but there is a problem. Unfortunately, glucose, amino acid, useful materials are dumped here. If you leave them, they go to the urinary bladder. We should not leave them. We have to absorb them. That means their dumping we have to show in the sequence. From the glomerulus, they dumped in the Bowman's capsule. Then after they absorbed into the cortical fluid, and then now they are taken out by peritubular net, the safe journey return back. Okay, chalo. Next, similar kind of sequence with, let's make it for A threshold substance. Means the substance which we don't require. For this, I am using excretory wastes here. We can say bile salts. We can say oxalates. We can say hippuric acid. These are some kind of excretory wastes which we don't want at all. Okay, so let's see their filtration. Actually, it should start the story from heart itself, left ventricle. Then from the left ventricle, left systemic aorta, dorsal aorta, then renal artery, then renal arteriole. Okay, renal arteriole, afferent arteriole. Okay, then glomerulus. Okay, so story we have to see here. This is the Bowman's capsule. Okay, then the blood vessel came here, afferent arteriole, glomerulus. Okay, then dumping is going to happen here. Dumping. Which one is dumped now? The so-called bile salts, oxalate, hippuric acid are dumped over. Okay, so where it come now? From there, where they are coming? From Bowman's glomerulus to, they came into the Bowman's capsule. Okay, then proximal convoluted tubule. Here you have to decide what I want to do here, what I want that I am going to do here, or what I should do. That suggest me. You should move forward. Actually, you should move forward. That because you have to eliminate this. If you go in this way, continuously, where you will go? Yeah, that means after dumping the waste material, we have to decide whether it is high threshold or A threshold. High threshold means we are absorbing. Okay, A threshold means we don't observe, we leave this. If you leave, where they go? Yeah, that's it. That means proximal convoluted tubule, descending limb of Henle's loop, ascending limb of Henle's loop. Okay. Then after distal convoluted tubule, initial collecting duct, and then straight collecting duct, then ducts of Bellini, calyces, pelvis, ureter, urinary bladder. Okay, so that's it. The ureter and the urinary bladder, because we have to dump this. Okay, so the same concept I'll show in the form of kidney, because the most of the times that we got questions from excretory system exclusively concept based, means we need to understand concept exactly. Okay, let me draw the kidney. First of all, let me draw the kidney. This is kidney. What are the blood vessels who are entering into the kidney? Who entered in the kidney? Renal artery. Okay, then who is coming out from the kidney? A renal vein and ureters. Okay, so this is ureter and this is renal vein. Okay, so now can I ask you directly here where the renal vein is going? What is their final destination? Actually, in circulation, we don't find final destination, but we are making heart as a destination. In that way, we have to say renal vein. Continue the sequence. Renal vein opens into post-cabal vein. Renal vein opens into post-cabal vein. Then heart. Can I call like this? Sure. Here you see the end point of this. Where does the ureter is going? Urinary bladder. Ureter is going towards urinary bladder. You people need to have clarity like this. So we have high threshold substances A, B, C. I'm writing here A, B, C. A, high threshold substances. Means here the substances which we require. Example, I'm writing glucose. Okay. Next, B, low threshold substances. Next, C, A threshold substances. Means here the substances which don't require at all. I'm taking example as uric acid. Some excretory waste it is. Okay, low threshold, interestingly, urea. We want little actually. Then now A, B, C entering inside. You see here A, B, C entering into the kidney. You are the people to decide over where does A should go, where does B should go, where does C should go. Written. This is written tickets. You have to catch two trains. One is the blue train, one is the green train. A, where the A should go? Yeah, A should go in this way because we want it. Means that should come out through the renal vein. And C should go in this way because we have to eliminate them. That's why C should go. B, we can say little here and little there. Means B, little in ureter and little in the renal vein. So this here it is renal vein only. Here it is only ureter. That is an interesting thing here. So exactly what is happening, if you understand, it will become so easy for us that what we are going to do. A sequence for high threshold, a sequence for A threshold. That means this complete journey in the form of sequences only. So you people, so actually if you are enough good white paper, if you are like this, there is no matter. You people will learn some about the kidney, some about the nephron, some about the renal artery, vein. Because of that only, I have to give enough time to you to sink this particular area in your mind. So that's why don't touch this area today. Just explain about the sequences. Just you people need to practice. So where I'll go today, where I'll go there, I'll take this item today. Ultrafiltration. We'll take this item, ultrafiltration. Okay, so we'll move on with the ultrafiltration. So how does the ultrafiltration occur? Today's topic is ultrafiltration only. Okay, this is a Bowman's capsule. Then who is our guest? Who is this? Afferent arteriole. When it enters inside, it is going to form a bunch of capillary network inside. What is that? Yeah, glomerulus. This is the red one, glomerulus. Then going out. Who is going out? Yeah, efferent arteriole. Do you find any difference between the diameter in afferent arteriole and efferent arteriole? Yeah, afferent arteriole diameter is comparatively larger if you compare with the efferent. Efferent arteriole to the afferent arteriole, diameter difference is there. Imagine in a closed bucket with a bigger pipe, I am dumping water. With a smaller pipe, I am taking water out. What will be the condition of the bucket inside? With a bigger pipe, I am dumping water, and with a smaller pipe, water is coming inside, large amount, and little is going out. So what can be expected inside? There a pressure is created. Okay, so now I'm going to say here pressure is created. More blood is coming, more blood is coming, more blood is coming, less blood is going. So that's why the blood capillaries of the glomerulus definitely are with some pressure. Okay, so that first we are talking about that particular pressure. The pressure created in the glomerulus is 60 mm of Hg. So how does the pressure is created? That we need to see. The diameter difference between the afferent and efferent arteriole. Afferent and efferent arteriole create the pressure in glomerulus. How much? Pressure created due to the diameter difference between the afferent arteriole and efferent arteriole in the glomerulus is, how much pressure is created? 60. Yeah, 60 mm of Hg pressure is created. So that means with that pressure, what we can do? Before that, with that pressure, what we can do? Before that, let me show you some two objects here. There is a brick and there is a bigger brick. So you are the kids. Definitely you might have did this particular. If you kick this brick with 100 ATP energy, by using 100 ATP energy, if you kick this brick, I'm not saying football, I'm saying brick. So brick will be dumped, that means will move forward till here. Okay. And the same person, same energy, with the same person, same energy. If we use 100 ATPs to push that he kicked this particular brake, can we say the same distance it will travel? No. That how much this is travel, the same distance it travels? No. Why? Yeah, normal language, you can say like that. Let us use some technical language. Opposing pressure, opposing energy. I'm using a word, opposing energy. Pressure we use there, energy let us use here. Opposing energy of an object is going to be, actually usually I use it to say this example with two types of human beings. I'll choose a thin boy in the classroom for the small break, and I choose myself here actually. Then, but we don't punch with leg. We will just push actually. Okay. So student giving punch like this is not a good thing. Okay. Chill. Let's talk about this. Whatever it is, whatever, if you push this person by using 100 ATPs, so he will move far. Then if you push this person with 100 ATPs, he may make some 2 steps a little backward, isn't it? Why? That means for every object, there is an opposing energy. Do you accept? Then just keep this in mind. Just keep this in mind about opposing energy. I'm using one more example, a hypothetical example, which is never possible. There, how many floors? 1, 2, 3, 4. There is a bigger tanker on the top floor. By using 1.5 HP motor, I am pumping water. That will go with some speed, isn't it? That will go with some speed. I am pumping water with some speed. The same motor, 1.5 HP motor, same motor, then same floor, same pipe, but I am pumping now blood. Sir, how you pump the blood to the 4th floor? Don't ask like that. Only answer I require here, this is water. It will go with some speed. This is blood. It will go with some speed. Then which one comes with more speed into the 4th floor? Motor is same. Water will come with more speed. Okay. Blood will come little bit slow, isn't it? Huh? I'm not asking about cotton and iron example you learned in 3rd class or 4th class. Think logically. Yeah, blood is somewhat viscous. Okay, if you are having problem with the blood, I do one thing. I pump pulpy orange here. At least that will come in the mind. Okay, I'll pump Sprite here. It's clear. Okay, now you say here, where Sprite will go faster or pulpy orange will go faster? Yeah, why? But because pulpy orange is little viscous. A lot of waste they dump inside the fluid. That is called pulpy orange. So they are saying this, claiming that it's clear. No, it's not clear. It is with carbon dioxide. It is with sugar. Lot of sugar, lot of carbon dioxide. But they are saying that it is very clear. Okay, choice is yours. No matter. Then okay, so that means you might have understand over blood is having some opposing energy pressure. Do you accept? Water also oppose. Motor is pumping up, water is opposing. Motor is pumping up, blood is opposing. Otherwise, how much energy we used over, that much energy result you don't get anywhere, any place. Okay, that means here I'll show you in the previous example what we said. By using 100 ATPs, I pushed a person. Okay, so there is a wall, he just hit that particular wall. Then by using 100 ATPs, when I kick him, just he moved a little 2 steps ahead, not reached at least to the wall. Okay, so let us describe this particular area. Let us describe. By using 100 ATPs, then he might have some opposed over. Do you accept here? At least thin boy, okay, but he'll oppose something or not? So he opposed some 20 ATPs. Okay. Then how much energy you used? How much energy you used here to push him? 100. How much he opposed?
20.
What is the net energy? 80. That's why he just hit the wall. Then here, he opposed some 70 ATPs, minus 70. Okay, how much energy you used? 100. How much he opposed? 70. Then just the 30 is the net ATPs that which moved him. Got my point here? For example, let's take one more example. There is a man, me itself. You are using 100 ATPs. With a great speed, you came and pushed me. I didn't move even. What happened? I didn't move even. You used energy, no doubt about that.
Opposing 100 force. 100.
Yeah, if it equals 100, you don't move at all. That means how much energy used over, that much if I oppose, I don't have any movement. Got my point here? Yeah, just keep all these things in mind. Now we are moving towards our topic. So how much pressure you created here?
60 mm.
How? Diameter difference. More is coming, less is going. Here pressure. Okay. With that pressure, they are trying to push the blood down. Okay. Don't you say that blood is also having some opposing? Don't they oppose if they push the blood? Now only we said, isn't it or not? Means with the, now only we said over, if we say that if you put some pressure, if you put some pressure, what happens if you put some pressure? That will oppose. Here also opposing. So I am writing here 32 mm of Hg, 32 millimeter of mercury. The opposing blood pressure of opposing blood colloid. Let me write the full name. Blood colloidal opposing osmotic pressure. We need to say. But you'll understand, no matter. Blood, due to their colloidal nature, it will oppose. With the help of 60 mm, you are pushing. Blood colloidal opposing osmotic pressure is 32 mm of Hg. Okay. And next one more, that is the fluid. Here some fluid is present. It is not empty vessel. Bowman's capsule is not an empty vessel. Definitely there is a fluid is there. Okay. So don't they oppose? Yeah, definitely they also oppose when you are pushing the blood down. They oppose. So that means Bowman's capsular opposing osmotic pressure is 18 mm of Hg. What is 18? Bowman's capsular opposing osmotic pressure. Bowman's capsular opposing osmotic pressure is 18 mm of H. Okay. Now you total the issues here. There, the diameter difference pressure created between afferent arterial and efferent arterial, diameter created pressure is 60 mm. Then blood colloidal opposing osmotic pressure is 32. And Bowman's capsular opposing osmotic pressure is 18. Now the total I want.
110.
Think little bit. This much number of examples I used. Think little.
Sir.
Thank you. Never I failed in this area. Never I failed. That means with the examples and illustrations that I don't put before people answer, I don't put minus here. But people will say that this time I appreciate the lecturer where he able to made the student to get answer 10 mm of H net pressure. Very good, and thank you to appreciate myself, given a chance to appreciate myself. So I think you got the clarity here. So that means diameter difference created pressure is 60. With that 60, you are trying to push down, they push up. How much they can create the pressure? 32. So you are trying to filter, make the filtration of the material. So that they also oppose. How much they oppose? 18. Then what is the net pressure? 10 mm of Hg. That 10 mm of Hg makes the filtration of the excretory wastes. Now let us name the topic. Ultrafiltration. This is called ultrafiltration. Just remember 32, 18, 60. Calculations are not permanent. That there may be a little vary in between, but definitely we need net pressure 10 mm of Hg. Net amount of pressure is required. With that net amount of pressure, filtration occurs. Okay, let us give the final conclusion here that how much blood is coming like this, we said from the heart? 1100 to 1200, isn't it? Which is 1/5 of the blood cardiac output. Okay, 1100 to 1200. Okay, let me say this once again. Let me say. So from the blood, ultrafiltration, just minimize. So how much is coming from the heart? 1/5 volume, 1100 to 1200 ml per day or per hour or per minute. Let me show you. Cardiac output equal to stroke volume into number of heartbeats per minute. Total cardiac output is 5040 ml or 5 liters. In the 5 liters, 1/5 we are sending to the kidney. From the heart, 1/5 we are sending to the kidney. That means this 1100 to 1200 ml definitely per minute is coming. Okay. And okay, whatever we created some pressure, because of the pressure, filtration is happened. Okay. Then we need to understand urine formation story with this. Water entered here. Okay. Among the water which entered here, we will discuss about glucose, amino acid, everything. But today we'll focus on the water. From the blood, water entered. RBC don't come. RBC won't come. WBC won't come. Nutrients came. Glucose, amino acid, water, all that came. And we have to start the work from tomorrow. Absorption of that, all that high threshold substances. But let us make water. Let us see the complete water. Among the particular water entered here. How much water entered here? That we have to see. That glomerular filtration rate in healthy individual is 125 ml per minute. In that, mostly water is only present. That's why I made water as a hero. 125 ml per minute. Okay. So that means per minute 125 ml per day. For every minute 125 ml. How much came here? Let me go in another page. Wait. Calculations seems to be little bit confusing. Okay. Now, what is the cardiac output? Yeah, from the heart 5000 ml, 540, no matter, 5040. Then how much enters in the kidney? 1100 to 1200 ml. Because of our effort, how much entered in the Bowman's capsule? Yeah, this is all calculations for per minute. All we are calculating per minute. Let me write here. 5000 ml from the heart, 1100 goes into the kidney, 1100 ml to 1200 ml goes into the kidney. In that glomerular filtration, GFR, glomerular filtration rate, we say 125 ml per minute. Okay. Just I am asking here, if you lose 125 million milliliter per minute, How much water you are going to lose in a day? 180 liters per day. If you have a little interest, you can calculate here also per day. But I am calculating here, NCERT calculated here. If you, in the same way, if you are losing water, 180 liters per day. If anything dumped here, what is their final destination? We made the sequence, urinary bladder. Sir, do we have 180 liters per day? Do we have 5,000? That means per minute 5 liter. That means per 1 hour. How much heart cardiac output for 1 hour? Did we calculate it? 5 liter per minute. 1 hour. That means
30,000.
60 minutes. 60 into 5 liter.
30 liters.
Yeah, 30 liters. Or we can say 30,000 ml. Do we have this much blood?
No, sir.
Yeah. If you want, you can calculate it for day also. 5,000 ml per day. That you can see that in lakhs, 1,40,000 something will come over. Okay. So, okay, whatever it may be. Here, if we don't absorb, what happens? You have to see. We have to absorb the water. How much we absorb? Along the length, we are going to absorb water. We are absorbing. How much we absorb? 99% we absorb. How can you say, sir? Generally, a human being will eliminate 1.5 to 2 liter urine a day. 1.5 to 2 liter urine a day. A particular person eliminate 1.5 to 2 liter urine. That means how much filtration is happening a day? 180 liters per day. In that, how much you are absorbing? 178 liters you are absorbing. How much you are leaving?
1.5.
Yeah, that means that's what simply calculation I said over 99%. Tomorrow we need to see here that how we absorb, what are the techniques we played for absorption, and what are the other issues is involved in the absorption. That all we need to discuss. That means we have taken a huge time for to get into the concept, to get into the topic. But that why? Because exact calculation required for us. I request the students to write about these calculations today. That calculation 60 mm of Hg, 32 mm of Hg, which we made some calculation. You should write something here. Okay. Then 18 mm of Hg, 10 mm of Hg, 5040 ml, what it is exactly, that we know cardiac output, 1100 to 1200 ml. Okay, then 125 ml per minute. All these are per minute only. That's the problem actually in NCERT. If you understand it as a minute, you don't get confused over. And 180 liters per day. 1.5 to 2 liters per day. Okay, 1.5 to 2 liters per day. So this is the topic we did exactly today. And a sequence from heart to heart we did actually in the previous class also. Heart to heart. And a sequence from Bowman's capsule to urinary bladder. These are the two things we did earlier class also. And now today, a sequence from heart to urinary bladder, I made it out. For whom it will come? High threshold, low threshold, A threshold. Yeah, A threshold. Okay, so that means here, by listening, you don't get anything. By listening, you don't get anything. Only you people will get the concept only by practicing. And our practice session should be vigorous in this particular area. If you don't practice, you don't get that particular concept. That means continuation. Continuation is so easy. Now here onwards, I just follow the NCERT text. Means here, to understand the NCERT text sentences, Every sentence have their own meaning and depth. So to understand them, this concept practice is very important.